So first of all, I would like to thank the chair for the kind introduction and also for inviting me to present our study on gigantism. And actually, Ireland, I think, is the best place for this talk because Irish history is full of legends about giants. So I have nothing to disclose. So pituitary gigantism is most frequently caused by pituitary tumors that harbor mutations in the AIP gene. And these tumors usually occur as familial isolated pituitary adenomas, or FIPA, or also in a sporadic way. Other monogenic diseases can also cause gigantism, for example, the McCune-Albright syndrome, or the Carney complex, or the MEN1. But most of these conditions occur in adulthood and in association with other tumors. In young children, somatic overgrowth due to growth hormone excess is very rare and so far of unknown etiology. So here are some examples of patients with pituitary gigantism. And so far, only about 100 cases have been reported in the medical literature. And here, I want to show you in the central panel, Robert Wadlow, also known as the Alton giant, that was the tallest man to ever live. And these pictures just point out that gigantism can also occur in families as part of FIPA. So the aim of this study was to discover the genetic defect causing the phenotype in patients with pituitary gigantism. And in order to do so, we performed genomic and genetic studies in 46 patients with gigantism. Patients were first screened for mutations and rearrangements in genes that are associated with pituitary tumors, for example, AIP, MEN1, PRKA-R1 alpha, and CDKs. But no mutations or deletions were found. Then some patients underwent whole exome sequencing and segregation analysis, and this led us to see the involvement of chromosome X. So we first screened a gene on the X chromosome, IGSF1, but the involvement of this gene in the phenotype was excluded. We then performed copy number variant analysis by employing different techniques. We used array-based comparative genome hybridization, or aCGH. We also used fluorescent in situ hybridization, or FISH, and we also employed individual TaqMan copy number assays. We then we performed sanguine sequencing on some candidate genes, and we also tested their expression by qPCR and immunohistochemistry. And finally, we also conducted in vitro functional studies in GH3, HEC293, and other cell lines. So first, by using standard aCGH, we found duplications in chromosome XQ to 6.3. And initially, we observed these duplications in 9 patients with pituitary gigantism, sporadic patients here, and also in 2 families. And recently, we also found other 3 patients with sporadic gigantism and with similar duplications. And if you look at the boundaries of these duplications, you can see that there are 2 regions that are shared by all duplications. And these regions are called smallest regions of overlap, or SROs. And in particular, you can see here, SRO1 and SRO2. And these 2 regions contain 4 protein coding genes. So with the help of Professor Lupsky at Baylor, we then refined our analysis by using high-density aCGH. On the left here, you can see some examples. So the red arrows represent the duplicated region. And underneath, you can see the sanguine sequencing of the breakpoint. And as you can see, the duplications are caused by very short sequences. Sometimes these sequences were homologous sequences, like in this case, or small insertions. And on the right, you can see a schematic drawing explaining the mechanism of these rearrangements. And as you can see, in some cases, these rearrangements were very simple tandem duplications. In other cases, the rearrangements were much more complex, like this one. And the mechanism causing these rearrangements was described previously and is called FOSTES, for fork stalling and template switching. And this is a replicative mechanism that can cause complex rearrangements of different scales. And in particular, FOSTES causes non-recurrent rearrangements, meaning that these rearrangements occur in multiple unrelated individuals with variable breakpoints, as we indeed found in our patients. And we also genotyped the duplications to confirm the inheritance pattern. On the left here, you can see some examples of sporadic cases, and as expected, we saw, as you can see, for example, here, that in the affected individuals, there is a positive band, whereas the unaffected relatives were negative. On the contrary, if you look here at this family, you can see that the two affected brothers inherited the duplications from their affected mother. So as I mentioned, we observed two smallest regions of overlap. In green here, SRO1, and in red, SRO2. So we then confirmed the aCGH studies by FISH. So we designed a green fluorescent probe for SRO1 and a red one for SRO2. On the left here, you can see a control sample. So this in blue, you can see the nucleus, and this is a female sample, so you can see the two X chromosome, and these are normal. In fact, you can see one red and one green dot. On the right here, you can see 4 sporadic patients with gigantism, 1 male and 3 females. In the male, of course, there is one X chromosome, and you can see that the chromosome is duplicated. In fact, you can see 2 red and 2 green dots. And if you look at the females, in particular, if you look at this one, you can easily see that one chromosome was normal and the other chromosome was duplicated. As I mentioned, in the smallest regions of overlap, there are 4 protein coding genes that are CD40 ligand, RGF6, RBMX, and GPR101. So we further confirm that these genes are duplicated by using individual TaqMan copy number assays. So if you look here, this is a control females, and you expect to see 2 copies, whereas you can see here that the female patients were found to have 3 copies. On the other hand, normal males having only one X chromosome, they are expected to have only 1 copy, but in these 2 affected males, we observe 2 copies. So this genomic analysis delineated 2 subgroups of patients, an early childhood form of gigantism that usually present in late infancy with XQ to 6.3 duplication, and a second form presenting mainly during adolescence, which does not have the XQ to 6.3 duplication. And I won't go into the details about the phenotypic features of these patients because tomorrow my colleague, Dr. Daly, will talk a lot about this. I just want to show you just one slide, just to show you the phenotype of our patients. And in particular, I want to point out, to show you the growth charts of these patients. For example, this growth chart here is for this patient here, this boy. And as you can see, as you can see, this boy started to grow rapidly when he was about 3 years of age, both in weight and height. And this pattern was also observed in a similar pattern was observed in all the other patients with gigantism. And also, in this case, this is a girl here, and again, here actually the rapid growth started even before, when she was 2 years old. And I also wanted to point out that these patients also have some acromegalic features. For example, you can see here large hands and feet. So we then wanted to test the expression of the 4 duplicated genes in the pituitary tissue, in pituitary tumor of our patients, and compare with controls, being both normal pituitaries and tumors, growth hormone-secreted tumors without the duplication. So for CD40 ligand, we did not observe any expression of this gene in any of the samples that we analyzed, whereas for both RGF6 and RBMX, we didn't see any significant difference in expression in the patients compared to the controls. However, for GPR101, you can see that there is a very high expression in the patients compared to all the controls. And we also confirmed this data at the protein level by immunohistochemistry. Here you can see in red, you can see the staining for GPR101, and in green, staining for growth hormone. And again, in normal pituitary or tumors without the duplication, you can see that there is no expression or very low expression, whereas we usually observe high expression levels in the patient with duplication. And also, if you look at the lower merge panel, you can see that surprisingly, GPR101 is expressed only in very few growth hormone secreting cells. We also wanted to see if this overexpression that we saw in the tumors was also present in other parts of the body, and so we extracted RNA from blood, and again we looked by qPCR at the expression levels of the 4 duplicated genes. But we were not able to see any significant difference, in the expression for any of these genes. Actually, if you look at GPR101, we actually saw that it was expressed at much lower levels than the controls. So we then wanted to see if GPR101 can also be involved in acromegaly. In fact, acromegaly and gigantism have the same pathogenetic mechanism, but differ regarding the age of onset. So we studied additional patients with pituitary tumors, and we screened them for copy number variants and for point mutations in the 4 duplicated genes. In particular, we screened 37 pediatric patients and 96 patients with sporadic acromegaly. And we did not observe any copy number variants, nor we observed mutations in CD40 ligand, RGF6, or RBMX. However, we found an interesting variant in GPR101, so we decided to sequence GPR101 in a larger international cohort of patients with sporadic acromegaly, and in particular, we sequenced 259 samples from 248 patients. In fact, for 11 of these patients, we had both germline and tumor DNA. And so we observed in GPR101 this missense variant, E308D, and this was identified in 11 out of 248 patients, mostly in tumor DNA. And this gives an allele frequency of about 2% for germline DNA and about 4% for tumor DNA. And in one case, you can see that this variant occurred de novo, being present only in the tumor DNA, but not in the germline DNA. And very recently, the exact database of controls became available, and actually this variant was found in controls, but with a much lower frequency that we observed in our patient population. So we then wanted to study in vitro the effects of the overexpression of both the wild type and the mutant GPR101 in growth hormone secreting cells, and we wanted to look at the cell proliferation and growth hormone levels. And as positive control, we used this mutation, A397K, that was not found in our patient, but was previously created and found and showed to lead to a 2-fold increase in the activity of GPR101. So we overexpressed both the wild type and the mutants, and when we look at cell proliferation, surprisingly there was no effects of the wild type. When we expressed the wild type, we did not see any increase in cell proliferation, whereas we saw a very modest increase with the mutants. But if you look at the growth hormone levels, again, the wild type did not increase growth hormone secretion, whereas a significant increase was observed for the 2 mutants. And then we also wanted to see if the overexpression of GPR101 can activate the cyclic AMP pathway. In fact, the involvement of the cyclic AMP pathway in pituitary tumorigenesis is very well known. So we employed a reporter, luciferase reporter, and we overexpressed again GPR101, and then we look at the cyclic AMP pathway activation both in basal condition and also after stimulation with phoscolin, that is a potent activator of adenylate cyclase. So as you can see, we did not observe any difference between the mutants and the wild type, but you can see that there's overexpression of the receptor increases a lot cyclic AMP pathway activation, so these receptors are high constitutive activity. So GPR101 codes for a G protein coupled receptor, and with the help of Dr. Costanzi from the American University, we built a predicted 3D structure of this receptor. And as you can see, the mutated residue is located in this intracellular domain 3 that connects transmembrane domain 5 to transmembrane domain 6. However, at the moment, there is no good template for this intracellular domain, so we cannot predict at the moment the effect of this variant on the structure of the receptor. So in conclusion, we described a new syndrome of early childhood onset gigantism that we call XLAG, and we found this syndrome in 18 patients with pituitary hyperplasia, or more commonly with growth hormone and prolactin secreting tumors. And this syndrome is caused by non-recurrent XQ26.3 microduplications, and we observed mostly in patients with sporadic gigantism, but this condition can also be inherited. And GPR101 was also found as the most likely disease driver in patients with gigantism, and we think that probably also in some patients with sporadic acromegaly. And we think that these results offer an opportunity to study a new pathway that is involved in the central regulation of human growth. And in the last few minutes, I just want to show you some unpublished data about the characterization of GPR101 expression in different species. So before, we observed that in mice, GPR101 is expressed in at high levels in the hypothalamus, in particular, as you can see here, in the arcuate nucleus. But when we look at the hypothalamus from human, however, we observed that only few cells were positive for GPR101. So we wanted to look in a panel of normal adult human tissues, the expression of the receptor. So first we did the screening by qPCR, and as you can see here, the expression levels were generally very low. So we then decided to concentrate on brain regions, and as you can see, the average expression is higher, and in particular, the highest levels were observed in the nucleus accumbens. So the nucleus accumbens is this region here that is located just above the hypothalamus, and that plays an important role as the reward center. And interestingly, about 25% of our patients also presented with an eating disorder. We also had access to human fetal pituitaries from autopsies, and we wanted to see if GPR101 is expressing during fetal development. And as you can see here, starting from 16 weeks of age, GPR101 is highly expressed in the human fetal pituitary. Then we look at the expression of GPR101 in the pituitary of different species. Here, I show you the expression in adult rats. In this case, growth hormone is in red and GPR101 is in green. And as you can see, most of the GPR101 signal comes from GH secreting cells. However, if you look here on the right-hand side, the other pituitary cell types are negative for GPR101 expression. This is in rats. Then we also looked at the expression in adult rhesus monkey, and we found different results because, as you can see here, GPR101 is not expressed in growth hormone secreting cells. However, it is expressed in gonadotrope cells. And finally, we also look at the expression levels during zebrafish development. So first, we did an RT-PCR, and as you can see, there is a bimodal GPR101 mRNA expression pattern. In fact, the mRNA levels wane during the first cell divisions, and these presumably represent maternal transcripts, and then gradually rise starting from the epiboly stage, and then stayed constant later on. And we also did in situ staining to see where GPR101 is expressed in the zebrafish embryo. So on the left here, you can see an in situ staining for growth hormone that labels very well the pituitary, and also on the right here you can see the expression of GPR101. That is, so the expression is is broad, and I think you can appreciate it more on below in the lateral view. So we decided to look to see if also the hypothalamus and the pituitary are stained by GPR101. So we cut this specimen, and here are some preliminary results showing that this seems to be the case. So this is the hypothalamus, and this is the pituitary, and there seems to be some positive regions. In the pituitary, there are some regions, but the staining is faint, so we are still working on this to see if this is real. So in summary, we saw that the brain is the major site of GPR101 expression across different species, although divergent species-specific expression patterns are evident, especially concerning the pituitary. In fact, in the pituitary, GPR101 expression is regulated in a cell type and developmental stage-specific manner in different species. And we think that this difference might reflect the very different growth, development, and maturation patterns among species. So we still don't know how GPR101 causes pituitary tumors and how GPR101 can cause secretion, oversecretion of growth hormone. In particular, we don't know if GPR101 is acting only at the pituitary level, if it is acting only at the hypothalamic levels, or both levels. So we are now developing some animal models to help us understand this, and we hope that these animal models will be like this. And finally, I want to thank all my colleagues in Dr. Startakis' lab, and also all our collaborators from all over the world, in particular Professor Beckers and Dr. Daly from Liège. Thank you.